Whew. Recording in progress.
Good afternoon. Hey. Okay, yeah, we're gonna do an activity session today, Tuesday. Activity session. Deep question 4 opens up tonight. Okay, so just a note on that, and then it's due Friday, right? So Thursday's class, same as usual. You'll have time to work on your deep questions and ask me questions if you have some. Okay. All right, so let's get started. This week was about transposable elements, about repetitive sequences and what they're used for. So you learned all about that in your pre-class materials. Okay, so today we're going to cover a few activities about chromosome copy number. We're going to go over some examples of whenever the mistakes happen and the chromosome copy number changes. Okay, so gene C is on a cat chromosome called D1. You have mated 2 cats. One cat has the genotype big C, CS. The other cat has the genotype big C, CB. Amongst their offspring is a kitten with trisomy of chromosome D1 that has the genotype C, CS, CS. So what does trisomy mean? Mm-hmm. Yeah, there are 3 copies of that particular chromosome. So this question is asking, okay, you got this kitten. It told you about the parents' genotypes, told you the kitten's genotype, the offspring. What parent did the kitten with the D1 trisomy inherit their C, big C allele from? Okay. 5, 4, 3, 2, 1. All right, so let's make sure we're having an understanding. 58% of the class is saying that the answer is parent 1. And where did that go? Oh. And 28% of the class are saying it's parent 2. And 15% of the class are saying you can't tell from the genotypes given. Okay, one other thing I'm going to tell you right from the beginning is yes, you can tell. You can. So let's go over this answer. Why did some of you say parent 1? How about you guys discuss this just for a minute or so, and let's do the poll again. Just take a minute, discuss with your neighbors, discuss with the people next to you. If you got a seat open beside you, you see somebody sitting by themselves, wave them over. Have some discussions. Right. Okay. So you guys discussed. Now let's poll again. Let's poll again. Go put your answer. Now that you've discussed with your neighbors, put your answer. So the answers changed. Let's see. 58% of the class were saying parent one. Okay, 5, 4, 3, 2, 1. Now 63% of the class is saying parent one. All right. Okay. Why? Why? Why parent one? I love that word. Yes, non-disjunction. One of those non-disjunction. But this, okay, so non-disjunction happened in one of the cats, right? Non-disjunction, we'll get to what that means in a second. Non-disjunction. Who can say, what's non-disjunction? Dude, okay. I'm just going to ask you from now on because you know.
It depends whether it's one like two, but it's an unequal
Right.
separation.
What's your name again? Christian. Yeah, Christian's got it nailed. It's an unequal separation. You know, whenever and it can actually happen during mitosis. That's actually common in plants. It happens in mitosis. Meiosis can also happen in meiosis. It's an uneven separation. You know how those chromosomes, DNA got replicated and it makes the X-like structure, right? And then the chromosomes, mitosis, those are going to separate. Meiosis, first you're gonna exchange maybe some crossing over non-sister chromatids. You're gonna separate the chromosomes first, and then you separate the chromatids next. Okay, so it's an unequal separation. Usually those chromatids, for example, they're supposed to go one in one cell, one in another cell. Chromosomes are supposed to split first into separate cells, and then the chromatids split into separate cells, and it's reductive division in meiosis. And so non-disjunction is whenever the chromosomes are not separating properly. Okay, so something weird happened here, non-disjunction. I heard non-disjunction. Okay, great. But what parent did the big C chromosome come from? Okay, so we have two parents here, right? One of them, they both got the big C, so that both of them can donate the big C. Only one of them has the CS, which ends up 2 copies of the CS. So definitely there's non-disjunction. It's got 2 copies of something you're only supposed to have 1 copy of in the first place. But so the one parent gave the CS. What parent gave the big C? Yeah, it's parent 2. It's parent 2. Okay, does everyone see that? Because they're each going to, it's not like all of the chromosomes come from one parent and none of them come from the other. No, this parent donated the CS. It actually donated too much. It donated too much. 2 of them got stuck together and it donated both of them, whereas parent 2 donated the big C. You see that? Okay, good. Okay, so if we had normal meiosis, this is what would normally happen. You know, you got your parent here, your parent cell. It's a germline cell. It's gonna, I don't know if this is the male or the female, it doesn't matter. So it's gonna undergo meiosis. That germline cell is gonna produce 2 gametes, right? These gametes actually they do have two copies. They each got a chromosome. That meiosis one gave them a chromosome. They're going to divide further. There would be further. This would separate into two individual chromatids would separate. But anyways, so ultimately the gametes end up with a single copy. That's what should happen. Okay, you could breed them together. This comes from parent one. This, if you got CB coming from CB is in the offspring, then the big C must have came from the other parent. Okay, is that clear? Clear. Okay, good. Okay, but in this cat, poor kitty, something happened during meiosis to produce gametes that were CS. So in other words, that the gamete didn't have that one copy like it's supposed to have. Something happened and it actually kept two copies. Uh-oh. It's not a haploid like it's supposed to be. It's a diploid gamete. Yikes. Okay, so and I'm gonna ask Christian, I'm gonna try to ask the rest of the class just this one time. I'm not trying to exclude you, but I just want to see if anybody picked it up. What is happening to give two copies of that CS? You're a great answer too. Anyone else? Anyone else? Yo. Okay, let's, what's the term? We'll get into the deets in a sec. What's the term? Aneuploidy describes a cell or organism that has more or less copies of a chromosome than they should, right? But what is the name of the, that's very good. You have to know that term too. But what happened? What is the event that happened to give rise to that aneuploid organism? Yes. Non-disjunction. Non-disjunction. Let's just, one word, non-disjunction. Very good. Non-disjunction. Everyone say it. Non-disjunction. Okay, good. I hope you're dreaming tonight. Non-disjunction. Non-disjunction. Okay, good. Now we got that term down. That's what gave rise. Look at this. This isn't right. This well, it's not right. It's not usual. This gamete actually has two copies. So it was non-disjunction, and the gamete. Thank you very much. I forget your name, but the term is it gives rise to an aneuploid. It's aneuploidy. Aneuploidy is the occurrence of one or more extra or missing chromosomes, leading to an unbalanced chromosome complement. Okay. All right. Disjunction refers to the separation of chromosomes and chromatids during meiosis. Non-disjunction is the failure of the normal separation process. Non-disjunction produces gametes with extra or missing chromosomes. So when did the non-disjunction event happen in parent one to produce gametes with the genotype CSCS? Discard C right now. You can figure this out. So C is not an answer. Please don't put C. And I'm going to give you a hint. If you draw it out, you're much more likely to figure this out than if you don't draw it out. Huh? What? Oh, okay. Okay. You guys are fast drawers. I didn't really see many people draw. But that's okay. We'll just go with it. If you drew it out, it would help make sense and you'd remember better. No? Draw it out. 5, 4, 3, 2, 1. All right. Okay, so I got 34% of the class saying meiosis one. It happened during meiosis one. I got 65% saying meiosis two, and I got 1% of the class saying there is no way, even though I told them that wasn't the answer. But that's okay. They clicked something, so I get the points. All right. So, okay, so did anyone draw it out? Maybe I didn't give you enough time. Yeah. Okay. So I would say like that's the way to check your answer. Like you can guess A or B, click, and then but if you draw it out, you that's the way you check it. It's like math. You can check your answer on this, right? Okay. So you can sketch it out. Let's go over it. I'm gonna do the sketching for you. So what do we normally separate during meiosis one? So here's our prophase. You had mentioned earlier, we got our chromosomes lined up. The purple ones are homologous, one from parent one from parent two, and the pink ones are homologous. They line up during meiosis one. What separates? We'll assume crossing over happens, and we're not really seeing the color changes here. Those very small regions, we just don't see it. So, what separates during meiosis one? Yeah. What? The homologous chromosomes. The chromosomes separate. Very good, very good. You got it. Meiosis one, chromosomes separate. What separates during meiosis two? Yes. Sister chromatids separate. Bam, got it. Very good. And okay, so let's see. Okay, so this was a homologous chromosomes aligned, homologs aligned. Here are so the homologs, they get separated. One chromosome goes to one end, one goes to the other end, and then down here. So this is homologous pairs of chromosomes. Here is sisters chromatids, and okay, so if you drew it out and you followed, like one of these is carrying CS and one of them's carrying big C, you would see ultimately what the difference is. Okay, so in parent one, we have non-disjunction. Let's pretend we have non-disjunction in meiosis one, okay? So what is the green one here? The green one is gonna represent C. Okay, remember this duplicated DNA. So both of these in the X, they both have the C allele, okay? And the gray CS. Okay, so now we have our meiosis one, and what's supposed to happen after meiosis one is, so first of all, these chromosomes are supposed to separate and go to separate cells, right? But they don't. If non-disjunction happens in meiosis one, they don't. And both chromosomes go to one cell, no chromosomes go to the other cell. It's uneven. You see that? So both of the chromosomes accidentally went here rather than splitting up green in here and gray over here. All right, so that's non-disjunction in meiosis one. And then what happens after that? Both of these would be pulled apart. Their sister chromatids would be pulled apart, and two of them would go into the cell. It's one from each would go into a cell, one from each would go into the other cell. Now, if that happened, what do we end up with? Do we end up with CS, or do we end up with big C, CS? Huh? Yowzers. You guys, this deep question might take you a little bit for a loop if you're not following along here. Yeah. CCS. I mean, you could just follow the colors and you would have the answer. It's CCS. This is C and this is CS. So in other words, if we had a non-disjunction event in meiosis one, you would end up with both copies, C and CS, that were found in that parent. That's not what we had. Remember, we had two copies of CS. Okay, so what happens if we have non-disjunction in meiosis 2? Well, meiosis 1 happens as it normally would. You know, you got some exchange, maybe what happened here. And then you got your homologous chromosomes being split up into separate cells. That's a reductive division. Then you're going to get another reductive division where the chromatids split into cells. So which one of these, the gray or the green, happen like they normally should? Green. Green. Okay, these guys are supposed to split apart. One homolog goes in one cell, one homolog goes in the other cell. It's a normal sperm cell or a normal egg cell. But non-disjunction in meiosis 2, so this doesn't separate in meiosis 2. And what happens? What do we get? We get 2 copies of CS in one cell, no copies in the other cell. Okay, there you go. So that explains what happened with this cat. Okay, this parent had donated 2 copies of CS. And so let's go back just for a second here. Remember our cat here, kitten, was big C, CS. This came from this parent, non-disjunction in meiosis 2. And this just came as it normally would. You got your big C copy from the other parent. All right. So before we start our activity worksheet, I just wanted to, I heard a lot in the answers. I scan them every once in a while. I got to check on what the TA is doing and how the students are doing. And I see a lot of language like swapping, you know, the chromosome swap. Chromosomes don't swap DNA. They undergo recombination. And it's not really a swap. It's a cut, right? There's a cut in the backbone and a linkage that happens. One chromosome gets to another during recombination. So this crossing over or recombination, it's a cut and a ligation. But anyways, so, you know, I just want you to remember, whenever we draw chromosomes like this, usually what happens is we'll draw it very simplified, right? Like we got these homologous chromosomes that align. And what's shown here is like these sister chromatids, this one here, and recombine with this one over here and vice versa, right? And so that can be resolved in different ways. But anyways, they're exchanging chromosomes. I guess a new concept that's being introduced here, I'll get into it more in a minute, but we have all these repetitive elements, right? These repetitive sequences, and they're located throughout the genome. It's not just on one chromosome. Other chromosomes have it too. And remember how the recombination machinery works is it looks for homology. And so if you have identical repetitive sequences on chromosome 19 as you have on chromosome 6, the machinery sometimes might make a mistake and think those two are homologs, and then it crosses over, transferring huge segments of the genome or of a chromosome from one chromosome to the other chromosome. Okay, so it's just a concept that's emerging. We're going to go into that in a little bit. Okay, so you have an activity, groups of two to three. You got this worksheet. I'm going to open it up. Follow the instructions, typical stuff. It is, I'll tell you specifically in a second. Oh. Okay. Okay, so week 9 in class, Tuesday, March 14 worksheet should be visible now. And you have the CrowdMart link. So I'll give you 15 to 20 minutes, okay? start our timer for 20 minutes.
Recording stopped.
Okay, we almost done. Okay, another 2 minutes or so. 4, 5, 3, 4 minutes. Mm. Okay, let's wrap it up. You guys can submit anytime for Wednesday.
Recording in progress.
All right, so woozits. Woozits are a diploid organism with 3 different chromosomes. So chromosomes 1, 2, 3. a zygote with a chromosome 3, so a zygote, chromosome 3 trisomy, so 3 homologs of chromosome 3, will not survive past gastrulation stage, so that's an embryonic stage. In other words, a chromosome 3 trisomy, so having an extra chromosome 3, is not compatible with survival to birth. Why does chromosome number matter? In this case, it's an extra chromosome number. I think it'd be easier. Okay, so let's do the flip side. What if we had one chromosome where we were supposed to have 2? What could be the problem? I'll give you a hint. Your explanation should involve some terms that you recently learned: haplosufficiency and insufficiency. Okay, so if you have you're short one chromosome, you only have one copy of chromosome. There's tens of thousands of genes, sometimes let's just say hundreds of genes on that chromosome. What could be the problem? I'm going to discuss with Christian in a moment, but before, yes. Okay, very cool. You like how you're using the term you said insufficient, right? Now, remember haplosufficiency or insufficiency that's every single gene we're generally talking about could be haplosufficient or insufficient. But in the case, you're absolutely correct, where you have a chromosome missing, so you only have one complement, you're supposed to have 2, and many of the genes are haploinsufficient. That means that one copy alone is not enough to provide the phenotype that you'd normally get from that gene. Very good. And if that's the case, you might not survive, right? The cells might not survive because of anything. Now, what about having an extra copy? Now we're not haplosufficiency, insufficiency. Never mind that, because we have 3 copies. Why might that be a problem? Yes. Okay. Very good. Very good. Is that what you were going to say? Okay, her answer was a little more clear. So you're saying you might have your gene product, protein, whatever the product might be, in excess. And in excess means you have too much of anything is not a good thing, right? We all know too much of even too much water. It's can be deadly. And so, yeah, so you have all these gene products and they're all supposed to do certain things. Remember, proteins, everything, operating in networks. These are complicated networks with proteins interacting with other proteins, doing particular processes. I'm just trying to think of an easy example. Nothing coming off the top of my head, but imagine you're supposed to have a certain number of proteins are supposed to form a network, a transcription factor network. And there's a certain ratio which they're supposed to physically associate to achieve a certain outcome, activate the expression of a gene just to the right levels. But if you have too much of one of the players, now all of a sudden the machinery is not fitting together properly. It's not being built properly. The gene's not being regulated properly that it's supposed to regulate. maybe not enough of that gene now because the parts are not in the right proportion. Driving a truck with 5 wheels rather than 4 might cause a problem. So that kind of idea, right? All right. Okay, so gene copy number is really important. This idea of dosage, it's actually very carefully controlled in cells, right? So more copies of a gene, more copies of its transcript, more copies of its product, ultimately can cause problems. And these can happen a number of ways, but trisomy is a good one. You know, you're only supposed to have 2 copies of the chromosomes. The Wu's had 3 copies of that chromosome 3. You can also have a copy missing, monosomy. You can have large scale gains. If you're in the plants class, we've talked about entire genome duplications. And plants are pretty special in that a lot of them can tolerate that. Bread wheat is like a hexaploid, and it had a lot of genome duplications in its history. Wide crosses, genome duplications, and bread wheat grows just fine. We're not all bread wheat. Not every chromosome can be like that. So, you know, sometimes it's really severe, sometimes it's not. So you can also have deletions, right, of large scale deletions. Now all the genes that were present right here on this chromosome down to 1 copy number. Or you can have duplications where part of the chromosome, your recombination didn't happen exactly in the particular loci that align perfectly. There was a little bit of a mismatch. So even though the sequences were the same, you can have a recombination event actually introducing more DNA onto a chromosome. Okay, and you can have inversions. Inversions are of little consequence oftentimes until you actually segregate the chromosomes out, and they recombine with a non-sister chromatid, and then the regions become duplicated or eliminated. And you can have translocations. You know, like I said, a part of a chromosome going to another chromosome becomes a problem because remember during meiosis, we're going to segregate one of each of these to one cell, one of each of these to one cell. And so we could end up with this region being in excess. Just this region might be diploid, might have 2 copies rather, and be abnormal. Okay, so here's another question you had. Alpha, who's a desert yeti, has a non-reciprocal translocation between chromosome 7 and 13. In the translocation, a part of the P arm of chromosome 7 has attached to the P arm of chromosome 13. So you got a piece of chromosome 7, now it's linked to chromosome 13. Yikes. The gene MSK is located in that translocated region. What gametes could Alphet potentially produce? So here's Alphet's 2 chromosomes. We're just looking at these 2. And so after translocation, this is what Alphet's chromosomes look like. And then those chromosomes are going to undergo meiosis. So which of the following gametes could Alphet produce? I draw it out. It's not showing up. Is it good? Is it good? You got it? Okay, 5, 4, 3, 2, 1. Let's see. All right. Very good, class. 53% of the class is saying E. Alphabet could produce all of these gametes. Okay, so keep in mind we're looking at two different chromosomes here, right? Chromosome 7 and chromosome 13. So this is not like showing you non-disjunction in that this is a chromosome 7 here and this is a chromosome 13, but there was a translocation event, right? So this chromosome here might pair up with this one. Pair up. It might go to the same gamete rather as this one here. All of these are possible. So all of these different combinations are possible. Very good. Alphabet had normal gene copy number, but gametes may not. Okay, so alphabet after this translocation, it's still, even though this just moved over here, was still normal copy number of MSK. There was still 2 copies as there was supposed to be. But now look at the gametes. Okay, just by that independent segregation of chromosomes, we got MSK being present here. It's not supposed to be here, and it's missing. And so you're only supposed to have 1 copy, right? But here you'd have 2. Over here you have 2, or I guess this one. Oh no, this one was truncated, right? This one was truncated. So here you're missing it here. So anyways, but you're missing some of the purple chromosome here. Here you have 2 copies of MSK and so on. So all of those possibilities are, all of those combinations are possible. Why might we see the following chromosome alignment during meiosis? So what was your take on that? These are 2 different chromosomes, not homologs. There has been an inversion of CD in the top homolog. There has been a deletion of CD in the bottom. Okay. 5, 4, 3, 2. Uh-oh. Okay. All right, so we got a spread, but most people are saying C. Let's start at the top here. These are 2 different chromosomes, not homologs. Why do we know that's not true? It's not true. Why is that not true? We got A, it's encoded on both. B is encoded on both. E is encoded on both. F is encoded on both. So chances are they're homologous chromosomes along the stretch. Now, let's look at B. What is an inversion? Yes, Christian. Very good, very good. Yeah, an inversion is whenever the order of, like A, B, C, D, the order actually gets flipped, right? So the chromosome actually got cut, flipped around, and it's back in its previous location, but it's inverted. It's so it'd be equivalent to being A, B, D, C, E, F, but that's not what we see here. We see it following it. It's telling us that the orders are the same. So the answer is C. There has been a deletion of CD in the bottom homolog. C and D are missing. They're missing down here. Okay. It could have been that this was an insertion. That wasn't given as a possible answer. Alleles with the plus have phenotypes dominant to those without. Okay, so the plus are dominant. What is the phenotype of the fly based on the figure below? Five, 4. This one, you should really wrap it. This is it's very straightforward, this question. 5, 4, 3, 2, 1. Okay. So, 40% of the class are saying B, 29% are saying D. Okay, so let's go over this here. So B is actually the correct answer. Okay, the phenotype Y plus is dominant to Y, CV plus is dominant to CV. And CT and SN, they have nothing to mask or cover them up. So their phenotype shows through. And then you have M plus. So B is the correct answer. CT, SN are hemizygous. Hemizygous. That means that there's only the one copy that they are. Okay, they're hemizygous. There's no identical locus on the other homologous chromosome. Okay, so they're unique. That site is unique. It's hemizygous. Okay, beta is a translocation heterozygote. Okay, reciprocal translocation between green and yellow chromosomes, I guess, occurred, right? How could beta's chromosomes align during meiosis? You would have to. You'd have to draw these out. So how would these chromosomes align in order to give us this particular result? Okay. And so note the blue and the yellow are non-homologous chromosomes, right? And so there's been a translocation event here where a part of the blue gets onto the yellow chromosome and vice versa. Just for these non-sister chromatids, okay? All right. Okay, so remember we're just showing the non-sister chromatids here. We're not looking at so when in terms of the actual genotype, we still have these guys here that are fully intact chromosomes that we're not showing. Now, okay, so we're zooming in on these non-sister chromatids. Meiosis, if 4 chromosomes, 2 pairs of yellow and 2 pairs of green have aligned as shown below, how do they separate? So that is, what are the gametes that result? Let me draw something here for you. I hope it'll help you understand this one. Okay, so whenever the chromosomes, they're aligning, right, and they're exchanging DNA, they've recombined. So remember I said recombination is a cut. So you have A, B, C, D. There was a cut here, and that got joined to the other chromosome, got joined at YZ from the yellow chromosome. And then you had VWX. Rather than have YZ, you have the EF that got joined here. This can separate in one of 2 ways, okay? It can separate like this, in which the D becomes attached to the E again, and everything is normal. The X gets attached to the Y again, and everything is normal. So in other words, we kind of cut this across like that, and the blue remains attached to the blue, yellow remains attached to the yellow. Okay, but if the chromosome cuts the other way, like this, A, B, C, D, Y, Z, okay? And then we have V, W, X, E, F. Okay, so that'd be another way that it would cut. So depending on how those get resolved, be determining whether you get this translocation event or whether it just goes back to normal. But the fact that these non-homologous chromosomes recombine in the first place, that's how you could potentially get the translocation event. Okay, so don't worry about this too much. I would never ask you a deep question where you had to like draw out the chromosomes and like that. This is really complex stuff. It's not easy to visually understand unless you really get into the details of drawing it out and doing those recombination events, trying to resolve the chromosomes different ways. Okay, so but ultimately this is what what you ended up with. You had your these homologs, they didn't recombine, and then non-sister chromatins, so to speak, that did recombine. Okay, so ultimately, this one here ends up with missing copies of E and F and 2 copies of YZ. See the 2 copies of YZ, E and F is gone. And this one has missing YZ, 2 copies of EF. Okay? All right. Okay, so in the last 10 minutes or so, let's just go over some stuff that you covered today in the class materials, because it's going to be on the deep question, so I want to make sure you're getting it. Whenever we have a reciprocal balanced translocations, balanced translocation is kind of like what we're showing here. They can often result in fertility difficulties. You can see here 7% result in infertility, 31% recurrent miscarriages, and so on and so forth. So they currently result in imbalance of the gene products, which ultimately leads to infertility. Okay, so now when we look at how the chromosomes or the genome is designed or is made up, it ultimately influences where and when where these things can happen, where these translocations can happen. Only 5% of the genome codes for proteins. By contrast, 50% of the genome codes for repetitive sequences. And you guys learned all about those different types of repetitive sequences. Remember, we broke them all down. And the pre-class videos was saying how the mini satellites, VNTRs and STRs are important. They're actually used in forensics to identify, blood, is this the blood of this person or that person? And is this potentially the killer's blood? Also, it's used to identify offspring, inheritance. Okay, and so how they do that is, you have these repeats all throughout the genomes, it's microsatellites, mini satellites, all spread throughout the genome. And they're essentially repeats, and different species have different numbers of repeats. Mammals have different numbers of repeats. Individuals in the Homo sapiens, so humans, we all have different numbers of repeats. It might not be, we might have the same at a particular locus, but the chances are that if you look at all 13 of a particular type of microsatellite, then if you look at all 13, no 2 of us in this room are going to have the same alleles at those 13 loci. And this is what the FBI uses in CODIS, the CODIS database. They have 13 of these STR sites. And what they do is they use primers. They digest first. I don't really know what the digestion, I think what the digestion is a technical thing so that when they do the PCR, it doesn't exhaust your DNTPs by going too far into the genome. It's going to stop where you got your digestion. But anyways, they use primers that flank these 13 sites. And the primers are essentially, they're going to amplify the site. And based on the size, they're going to know how many repeats are there. Okay? So they could distinguish one individual's allele from another individual's allele. So you run a gel for an hour. As the gel runs, you may see multiple fragments with one lane, and these begin to spread out. What is the difference between a fragment that is closer to the positive end of the gel compared to the band that's closer to the negative end? Negative end, positive end. Which one's going to have the larger size? Okay, 5, 4, 3, 2, 1. Those of you working in a lab, you probably get this right. So 64% of the class is saying A. The band closest to the positive end represents the smaller piece of DNA. Okay. And that's correct. The reason why that's correct, DNA is negatively charged and it's getting pushed through the gel from the negative end to the positive end. And the bigger DNA gets slowed more than the smaller DNA. And so it gets slowed down, and ultimately it comes out to individual bands like this. This is thousands and thousands of pieces of that big DNA. They just migrated at the same rate. They were put in this well. They migrated at the same rate and they end up in the same place and they make a band. This one down here would be smaller. This one up here would be larger. You stain your gel so that you can see the DNA bands. Why do you see two fragments in lanes 1 and 2 indicated with blue arrows that differ in how bright they appear? Why do you see difference in brightness? Five, 4, 3, 2, 1. Okay. 56% of the class saying B. There is more DNA in lane 1 than there is in lane 2. That is correct. Okay. Difference in brightness. This reflects how many PCR products are there. If you have more copies, you have more DNA. So this one might be twice as many copies as this one here. We're almost done. Last slide. You have genotype of polymorphic STR and chromosome 3 for your family with 3 children. Given the results below, which of the genotypes in the list below A to D does not represent the children of parents 1 and 2? These numbers are referring to these sizes of bands. Okay. You want to understand this one. 5, 4, 3, 2, 1. Okay. Let's see what we got here. Oh, we got a big spread. Okay, got a big spread. 36, 28, 18. Okay, so 36, the highest, the most chosen answer was C. Okay, so let's go through this very quickly. Okay, so is A one of the, this is a parent. These are the 2 alleles that could be given. And this is the second parent. These are the 2 alleles that could be given. Now we look at A, 154, okay, and 146. Okay, that's essentially child 3, right? And so they've got 1 allele from here, 1 allele from here. Great. Let's go to C, because that's the one the class said the most abundant, most popular answer. 150, 146. 150, okay, 146. That's one of the children right there. 150 came from this allele from this parent. 146 from this parent. What about D? 150 and 140. That's a child too. The answer is B. The answer is B. Why is the answer B? 154 to 140. I know it's a little bit tricky. 154, 140. It's possible, but no children on the list have that genotype. Wah, wah, wah. It's a little bit of it's a possibility, but you had to look. Those are the 3 children. That one wasn't there. Okay, guys? Very good, very good. Okay, remember, deep question comes out tonight, due Friday, Thursday, working in class. I'm here for questions if you have any. Okay, please ask, do great in the course. That's my pep talk. Thank you.